Good afternoon to everyone. Thank you for joining this week's seminar from the Living Earth Collaborative and Washington University's Evolution, Ecology, and Population Biology group. Today, I have the great pleasure to introduce Dr. Elizabeth Carlen. She is a National Science Foundation and Living Earth Collaborative postdoctoral fellow working with Dr. Jonathan Lossos at Washington University in St. Louis and with Dr. Chris Skell at the University of California. And it's important to mention that Dr. Carlen is one of the new Living Earth Collaborative postdoctoral fellows of this year. We are fortunate to have you here, Dr. Carlen. Dr. Carlen earned her PhD degree in biological sciences from Fordham University in New York City in the Moonshine South Lab. Her work was focused on how urbanization affects the evolution of feral pigeons in the Northeastern megacity, from Boston to Washington, D.C. Dr. Carlen is building an impressive profile of publications in prestigious journals such as Animal Behavior, Ecology and Evolution, and Evolutionary Applications. Her work brought the attention of the media with broad coverage in newspapers, magazines, radio interviews, podcasts, and documentaries. For instance, her research was featured on the Saturday Night Live show and led the New York Times to refer to her as the pigeon stalker. Because she is a vivid outreach person, she is co-founder and editor of the urban evolution blog called Life in the City, Evolution in the Urbanizing World, where you will find interesting thoughts about animal evolution in urban areas. Her research is centered on the influence of humans in the distribution, genetic diversity, and the interactions of the species in suburban and urban ecosystems. And her postdoctoral research is focusing on the impacts of urbanization and environmental racism in Eastern great squirrels. One of the funny facts that is that Dr. Carlen does in her free time, she looks for roadkill animals to prepare them for educational purposes. If you find some, please contact her. She will be happy to have them as part of her collection. Before I hand it over to Dr. Carlen, I want to remind all the audience to leave questions and comments in the YouTube chat section, and we will read them at the end of the seminar. And stay tuned for the next week's seminar by Dr. Fernando David Najera Munoz. Without further delay, let me introduce you to Dr. Elizabeth Carlen and her presentation entitled From the Ground Up, Urban Evolution in Squirrels and Pigeons. Take it away, Dr. Carlen.
Thank you so much, William, and thank you everyone for showing up today or watching this later on YouTube. So I am new to St. Louis, and what I'm going to be talking to you about today is kind of some of my dissertation work and then what I'm planning on doing during my postdoctoral research. So as we know, urbanization is increasing drastically around the world. And in this video here, what you're seeing is this massive land shift in China over a course of 30 years, where not only is this urban center building out and connecting with this other center, but also that we're getting just this build out into the ocean and this drastic kind of increase in impervious surface as the land is converted from these natural areas to human used environments. And with that comes increased habitat fragmentation, as we can see here in San Francisco, where places like the Presidio of San Francisco up here and Golden Gate Park are really isolated from each other with only this small strip of land, green space connecting them. And so you can imagine being an animal such as a fox or a coyote trying to transverse San Francisco and not having a lot of space to do that, or a lot of green space to get between these areas. We also see increased buildings and roads, just massive impervious surface. and increased air, noise, and light pollution, as we can see with this smog that's going on here, and increased environmental toxins. So just an increase in kind of how the earth is used and what is being put into that earth. And with these massive landscape changes and environmental changes, we get a new ecosystem that has novel selection pressures and barriers to gene flow. And the ecology of this urban environment drives evolution, which in turn then influences the ecology. So I'm really interested in these processes and how they're influencing each other within urban spaces. And urban evolution studies are really kind of this new area that has really only taken off in the past about 20 years. So it might be surprising that despite the fact that we've been living in these urban spaces for a really long time, that we only began studying them and publishing on urban evolution in around the mid-1990s. And as you can see, there's just been this giant explosion recently of urban evolution papers. And this is only taken off further as those of us that have recently gotten our PhDs are finally publishing our dissertation work and really focusing our entire careers on urban evolution studies. And to show you the breadth of this work, these urban evolution studies have focused on a variety of taxa around the world and mainly looked at one or maybe a few cities to compare evolutionary phenomenon. So here we're seeing in black silhouettes of the organisms that have been studied, and in blue, human commensals that have been studied, which with each colored dot representing a city and the size of the dot based on how many people are living in that city. And these studies have focused on a variety of evolutionary phenomenon, including morphological changes and physiological changes, behavior, gene flow, genetic drift. So kind of encompassing a whole bunch of different things. For example, urban birds are derived at the CERT gene, which was associated with harm avoidance behavior. And former WashU postdoc Kristen Winchell showed that lizards in Puerto Rico have longer legs and stickier feet that allow them to move more easily on urban substrates. For example, if you're a lizard that is in a very open area that doesn't have a lot of trees and a tree is your refuge, you basically need to be like Michael Jordan to get across that urban space and have really long limbs to be able to traverse that space quickly. And then you need those sticky feet to cling to areas like what this lizard is doing, hanging out on painted metal, which is a much different surface than that tree surface that lizards are normally found on in natural areas. And a lot of urban evolution studies have looked at adaptation, but another consequence of urbanization is alterations in gene flow. For example, the northern house mosquito colonized the London underground and subsequently diverged into separate forms that are genetically and behaviorally distinct from the above ground form. And this is because their behaviors can be different because there's constant lighting in the underground, and the time of day that people are active are going to be different than above ground. There's going to be these huge rush hours in which more people are going to be on the subway, which the mosquitoes can feed on. So this last story provides this example of non-adaptive evolution in which gene flow and genetic drift work to counter each other, with gene flow homogenizing allelic variation and genetic drift differentiating allelic variation. And we see different effects of drift on genetic structure of populations that are dispersed across the urban landscape. For example, with bobcats, we see that urbanization, specifically the building of freeways here, like the 405 and the 101, have really led to genetic differentiation and genetic drift within the population. So what you're seeing in this image is each circle represents an individual bobcat sampling location, and the colors correspond with the predominant structure or ancestry cluster assignment for each individual. And you can see that here along the 405, we have to the right of 405, we have this yellow cluster, and to the left of the 405, we have this purple cluster, and to the north of the 101, we have mostly green individuals, some red individuals. So we're getting different individuals kind of separating out along these highways, presumably because it's very difficult for these individuals to cross these highways. But another consequence is in black widow spiders. Lindsay Miles showed that urbanization actually facilitated gene flow, likely because these black widows are building their webs under cars, and then these cars are traveling between cities. So these cars people are more likely to move between urban space and urban space than between urban and nearby rural space. And because of that, the widows are being transported along these urban transportation routes. And so she found that these black widow spiders were more similar among these urban areas. Now, these are examples of native species, but gene flow and drift can have varied effects in organisms that rely on humans, which I'll refer to as human commensals. And human commensals are species that live in close association with and benefit from humans. And often these organisms are so integrated into our cities that we don't even notice them. And most urban evolution studies have really focused on native organisms. However, there's a lot to learn from organisms like human commensals that not only persist, but thrive in urban areas. And of course, our understanding of spatial genetics of human commensals is really dependent on each organism and their relationship with humans. So just to kind of point out, you probably recognize a couple of these human commensals: the house mouse, the cockroach, the rat. This includes brown rats and black rats. We have bed bugs here, mosquitoes, we have lice, and then of course the pigeon, which I'll talk about a little bit more in a bit. So, for example, in German cockroaches, which are a human commensal, researchers found that genetic differentiation increased with spatial scale and was lowest within a building, meaning that all the individuals within a building were highly related to each other. And as you got further away from the building, the individuals were less closely related. So this kind of makes sense, like, you know, your family unit is living within that building, and as you move out, individuals are less closely related. And my lab mate and academic sibling, Dr. Matt Combs, showed that in brown rats, there was limited dispersal in New York City, in the island of Manhattan in New York City, that led to uptown and downtown genetic clusters of rats. And he spent a long time trying to kind of figure out what was going on here, and realized he had a very difficult time trapping in kind of this midtown area of New York City. And you can just see that his sampling is a lot less dense in this area. And it wasn't for lack of effort, it was just simply harder to catch rats in those areas, possibly because there's a lot less rats there. And this, when he thought about this, it really seemed like this area has a lot less people living in it, and maybe therefore a lot less trash and less food for these rats to feed on and sustain their populations. And so that was kind of forcing these uptown and downtown genetic clusters, was kind of this barrier to gene flow here across Midtown. So I kind of wanted to come into this and ask, but what about pigeons? And we know that spatial scale is really important and needs to take into account life history strategies. We all know that pigeons are capable of flying super long distances, and there are stories of pigeons heroically bringing messages back to bases during World War II. However, GPS studies have shown that pigeons travel on average about half a kilometer from their roosting location, because why would you leave home when you have plenty of food and a nest and a mate? So I began to kind of wonder, with pigeons, will I see similar genetic patterns of those observed in cockroaches, where there's high similarity among individuals and a shared geographic location? Or will I see something along the lines of rats where there's these distinct genetic clusters? Or will I see something totally different? And to kind of give you some background on pigeons, I'm guessing everybody in the audience has seen a pigeon and interacted with a pigeon, but just to give you some background on these individuals, pigeons are native to Asia, North Africa, and Southern Europe. And they were introduced to North America when colonizers came here. And they were originally domesticated around 5,000 to 10,000 years ago as a food source. And they not only provided a food source, but their guano or their feces was very rich in nitrogen, and so could be used to fertilize soil. But as other poultry became more popular, there's a lot more meat on a chicken, pigeons became less favorable as a food source, and people began breeding pigeons for their show traits. As you can see here, this individual has these curly feathers and this kind of distinct coloration, this reddish brown, and their racing abilities, so their ability to fly back and navigate home very quickly. And they're known for their long distance flight ability and their ability to return to their home loft. So pigeons are excellent flyers and very good at navigating. And they also have an incredibly rapid reproduction and fast population turnover. One of the questions that I constantly get is, why have I never seen a baby pigeon? And one reason is they're not particularly cute. You might not be searching one out. They're as you can see in this image here, they're not particularly adorable. They look like kind of creepy dinosaurs, and they stay in the nest until they're almost full grown. So about every 6 weeks, a clutch, a male and female, can produce a clutch of pigeons. Typically, there's 2 offspring in that clutch. And then these 2 pigeons will stay in the nest until they're almost completely full grown. And pigeons have been used as a model of evolution, from helping Darwin understand selection to Dr. Mike Shapiro's lab at Utah that is working on determining the specific genes involved in many of the fancy traits that we see on pigeons, such as head crests and feathered feet and curly feathers. And to give you a little bit of what we know on pigeon population genetics, studies in Europe have shown that genetic distance increases with geographic distance, and that inbreeding may be common within flocks, and that on average, pigeons are making daily flights of about 590 meters from their home roost. So they're traveling about half a kilometer. And in Singapore, we know that pigeons form a single genetic cluster, but it's unclear if this is because of the island nature of Singapore. Singapore is an island country and is a rather small country that's isolated on this island. And pigeons were introduced to Singapore in the 1960s. So it could be that there just hasn't been enough time for pigeons to diversify on this island at this point. So I'm going to share with you today kind of 2 overarching questions for my dissertation. I don't want to present everything that I'm doing here, so just sharing a couple of things. So first, asking, how do gene flow and genetic drift structure the feral pigeon population in the Northeastern United States? And how variable is flight initiation distance, or how close you can get to a pigeon before it moves, along an urbanization gradient in New York City? So first, jumping right into this first question, I wanted to look at originally just New York City, and my PhD advisor was like, Liz, you need to think about pigeons and how far they can fly and their ability to disperse, and really kind of think about the larger scale. And what's really interesting about where we were located in New York City is that New York City is in the middle of this northeastern megacity. And this northeastern megacity covers less than the 2% of the nation's land area, but contains over 52 million residents, or about 17% of the US population, making it the most heavily urbanized area in the United States. And most urban evolution studies thus far have really focused on a single city or done comparisons among a few cities. But Obviously, animals don't respect political boundaries, and the extent of urbanization throughout the Northeast, along with the dispersal ability of pigeons, makes the Northeastern megacity really an interesting case study for urban evolution. So just to familiarize yourself with this area, it ranges from Boston up in the north down to Washington, D.C. And I focused on 6 major cities, as you can see starred here. And when I pull away these stars, if you look at these red spots, this is showing impervious surface, and each one kind of looks like it could be almost an island separated by green space and less developed area. So my first thought was kind of, okay, well, maybe each city acts as this little island, and this is an archipelago of islands in which the pigeons are isolated from one another. But then when you look at the same area at night, what you see is just massive amounts of urbanization, where almost from New York City down to Washington, D.C. is almost one massive urban area. And then we get some slight kind of separation across Connecticut here. And then Boston and Providence again become this interconnected, almost single city. Okay, so that kind of starts to inform my thinking about what might be going on. And with that, I want to kind of talk about some possible spatial population genetics models. For example, we might have panmixia, where each individual is equally likely to breed with another individual. We might have isolation by distance, where you get genetic differentiation over geographic space. We might have isolation by barrier. And typically in urban areas, we're thinking of isolation by barrier as something like a road, like we saw in those bobcats, where the bobcats couldn't cross the road. And so we saw differentiation on different sides of the highway. But with pigeons, I want you to think about if you've ever seen pigeons deep in forests or deep in suburban areas. And so I kind of started to think about, well, maybe these forests or these suburban areas might act as a barrier for these pigeons, where you're getting population differentiation on each side of that forest barrier. So I went about collecting pigeons in each of these cities, up and down the Northeastern United States, and ended up with 473 individuals in my sample size, or in my sample. And just to kind of give you an idea of how I went out and collected pigeons, I'm going to show you a video here of me catching pigeons. So this is what I used. It's called a net gun, and it shoots out a net over these individuals. I've laid down some corn seed and then shot this net over them. And while you can see a bunch of them get away, we caught 10 in this net, which was more than enough for a sample size in this specific location. I didn't really need more than that for this location. And then once I had these guys sampled, I took them back and took them back to my car, which I had set up as a mini laboratory, field laboratory, and collected blood for DNA analysis, and then did ddRAD, or double digest restriction enzyme associated DNA sequencing. What I want you to know, if you're not familiar with ddRAD, is it's reduced representation genome sequencing, where we take enzymes, here shown like in red, that chop up the genome so that we could extract thousands of small regions within which we can look at relatedness of hundreds of individuals based on single nucleotide polymorphisms or single base pair differences. So it's a way to get representation across the genome without sequencing the entire genome. I then use Bowtie to align these sequences to the genome. We have a pigeon genome, which is great, and stacks to identify SNPs, before moving on to my analysis, where I retain 35,000 SNPs. So pretty good sample size. And this was, remember, 473 individuals. So kind of zooming in and what I found. So first, to look at relatedness, I want to show a Mantell correlogram. And so what you're looking at here is distance across the x-axis and relatedness across the y-axis. And a correlogram is a graph in which spatial correlation values are plotted as a function of geographic distance classes among the study sites. So what this is showing me is that pigeons within a 50-kilometer radius are highly related. And if you think about the average size of my cities, kind of minus New York, most of my cities had a radius about 50 kilometers. So it makes sense. All the individuals within that city are highly related to each other. And beyond that distance of 50 kilometers, pigeons were unlikely to be related to each other. So pigeons are unlikely to moving outside the city, but it only takes one migrant per generation. So let's run some other models. Let's run some other tests to see what's going on. I next ran EAMS, or estimated effective migration services, to look at fine scale patterns of gene flow between cities, specifically because there may not be uniform processes that are leading to genetic clustering in such large areas. And EAMS is able to highlight this variability. And we found that we might be seeing barriers to gene flow between cities, which may not be detectable by other analyses. So just to kind of orient you with this image, our null model is isolation by distance, and that's represented by white. In blue, you're getting higher than expected gene flow, and in red, you're getting barriers to gene flow. And I've circled kind of our sample locations here in black, and what you're getting is basically isolation by distance, or maybe a little bit higher than expected gene flow within our sampling locations. And then kind of this barrier to gene flow a little bit between the cities. Now, EAMS is very sensitive to overinterpreting data where it doesn't have any information. So as you can see, all to the west, it has this higher than expected gene flow, and it's just overinterpreting there because it doesn't have any sample locations there. So EAMS can be really helpful if you can kind of zoom in and look at what potentially is going on, but needs to be interpreted and taken with a grain of salt and taken kind of with some other analyses. So what I'm seeing here is that there may be some fine scale structuring that's going on within the cities, and some barriers to gene flow, and that would confirm what I saw in my Mantel correlogram. So again, we're seeing that potentially there's some barriers that are happening between the cities. Now, when I ran admixture to look at relatedness among cities, and to orient you, those of you that are unfamiliar with admixture plots, each individual in this plot is represented as a single bar, and the color represents the genetic cluster assignment for each individual, displayed as proportion of ancestry. And the K value represents the number of populations. And when I ran the analysis up to 15 populations and then compared my models, K equals 2 was always my most well-supported K value. So meaning that I had the most support for 2 populations. So what I'm seeing here is that we have mostly individuals from Washington, D.C. through New York City forming one population, and that our individuals from Boston and Providence are forming another population. Okay, so they're not individual islands that when we run this admixture, that we're actually seeing that there's actual movement happening between New York City, Philadelphia, Baltimore, and Washington, D.C. that's really connecting these populations, and that there's not really movement happening between the Boston and Providence individuals and cities that are further south. And to kind of confirm this, I also ran a DAPC, or discriminant analysis of principal components, that transforms the data using a PCA before discriminant analysis to ensure that the variables submitted are uncorrelated. And DAPC structures groups so that within group genetic variation is minimal while maximizing diversity between groups. And what you're seeing here on these axes explains 93% of the variation seen in these data. So again, kind of looking first across this x-axis, what I'm seeing is that Boston and Providence are pulling out as separate from our other cities. And then when we look along our y-axis, we're getting separation of Philadelphia and New York City and Washington, DC, and Baltimore. So overall, with this, what this is telling me is most pigeons are not moving outside the city, but because a few individuals are dispersing, we're seeing this unrestricted gene flow between most cities within the Northeastern United States. And however, there's this suburbanization across Connecticut that may be acting as this barrier to gene flow, as we saw in that picture of the light at night, where that suburbanization between Boston, Providence, and more southern cities really prevented possibly this kind of overall connectivity of these cities. Next, I want to tell you about flight initiation distance, or how variable this flight initiation distance is in New York City. And flight initiation distance is a common measure used to assess weariness and risk, and is defined at the distance at which an escape response is initiated between a threatening stimulus and an animal. Basically, everybody has probably done this study. So everybody has approached probably a pigeon at some point in their life and seen how close they could get before it flew away. And kind of anecdotally, this study came out of people saying to me, well, why can I get so close to pigeons in Times Square, but not get close to them in the outer boroughs? And so I wanted to look at the relationship between FID and landscape features, and also the predator community that is in New York City. And I have to give a huge shout out to Richard Lai, who conducted this research as part of his senior thesis while he was a student at Columbia University. Richard's now a graduate student at Yale University in the biology department. So this is what he did, kind of walking at that pigeon and seeing how close he could get before it moved away. And he repeated this experiment 519 times all over the city. And then we use these data to look at a bunch of different variables. And I wanted to kind of highlight how heterogeneous the landscape is in New York City. I think oftentimes when we think about New York City, we think of some place like Times Square that is very, very urban and full of tall buildings. But here we see Times Square that's, yes, highly, highly urban, but there's these giant fields in Central Park. There's these cemeteries out in Queens. There's these very suburban areas out in Jamaica Beach, and industrial areas along Newtown Creek, and even heavily forested areas within Prospect Park. And so the landscape across New York City is very heterogeneous, extremely varied. And there were areas, while there are pigeons in most of the places across New York City, what we ended up finding was that there were plenty of places that didn't have pigeons. And in addition to places not having pigeons, we wanted to test if we could get closer to pigeons in these different areas that did have pigeons. So we collected a bunch of different landscape features and put them into a PC analysis to reduce dimensionality of the data, and then use them in subsequent analysis to build our model. And so what I want you to focus on here is that you're seeing both color and size of the circle, indicating how the variable is associated with each principal component. So focusing in on just impervious surface, we see that PC1 is positively associated with impervious surface. PC2 is negatively associated with impervious surface. PC4 is slightly positively associated with impervious surface, and PC3 is really not impacted by impervious surface. So we repeated this for a bunch of different landscape variables, such as canopy cover, developed low intensity land, developed medium intensity forest, herbaceous plants, water, road length. and put them all into our model. And then we put this in along with pedestrian traffic. One of the great things about being in New York City is there are lots of data about just the city in general. And one of those data sets included just where pedestrians are. And this was part of originally a business atlas data set where to encourage businesses to move to certain areas of the city because there's lots of foot traffic. And so kind of taking that data and plugging it into this landscape model about where pedestrians were, and then along with human population density. And I wanted to look at these two separately because in New York City, there are many places that have high pedestrian traffic, but low human population density. Thinking back to Times Square, there are lots of people that pass through Times Square every single day, but do not live there. And you can think about that in other cities. There are places like malls or airports that have lots of pedestrian traffic, but nobody living there. And so while human population density is often a good proxy, there's something to be gained by also adding pedestrian traffic into that model to kind of get a more precise picture about what might be going on. So what I found, as you might expect, is that you can get closer to a pigeon in areas where more people live. You can get closer to a pigeon in areas with more pedestrians. And you can get closer to a pigeon in areas with more impervious surface, more roads, and less canopy cover. So this kind of all made sense, but we were the first ones to test it in pigeons. And I think this is kind of one of those nice little studies where you have this kind of anecdotal saying like what's going on here and going out and actually testing that natural history or that observation. And then we also wanted to look at predators because predators obviously are in the city of New York. And while we can think of lots of predators that are potentially influencing these pigeons, we decided to focus on avian predators. And because raptor occurrences are relatively low and time consuming to collect, I turned to community science data collection using the app eBird. And since I only wanted to understand if there was a correlation with predator sightings, such as abundance and presence, I used counts of raptor sightings within a 590-meter radius of each observation. And that's, again, based on that distance on what an average home range is of a pigeon. And similar to the landscape model, I ran a backward stepwise model to determine which model was the best fit. And include my covariates, as well as the number of red-tailed hawk sightings, peregrine falcons, and Cooper's hawk sightings. And what I found was that Cooper's hawk sightings have no influence on FID. And that FID, flight initiation distance, increases as red-tailed hawk sightings increase. So you can't get as close to a pigeon when there are red-tailed hawks in the area. And flight initiation distance decreases as peregrine falcon sightings increase. So what's going on? Why are these two so different in kind of their responses? What would possibly be going on here that pigeons would potentially be acting differently when different predators are around? And this really kind of confused me for a long time. And I spent a lot of time talking to naturalists. And just to kind of show you, this was definitely what our data was showing us. We ran this multiple times. Here's a path analysis on the reduced, most well-supported model. And again, it's telling me the same thing. And that red-tailed hawk sightings positively influence FID and peregrine falcons negatively influence FID. And so kind of trying to figure this out, we were really struggling and talking to ornithologists and kind of came up with, well, maybe something is going on with how these different birds hunt pigeons. So what you're seeing in this video here of a peregrine falcon hunting is it's grabbing the pigeon out of midair. So this peregrine falcon is going to kind of dive down. They're known as sky Lamborghinis. They can go incredibly fast. And it's going to dive down and grab that pigeon while it is flying. Whereas a red-tailed hawk is much more of a ground predator. So this red-tailed hawk is going to dive down onto the ground and grab that pigeon while it's on the ground. And if you're an unfortunate pigeon that gets caught on a balcony, and happens to be a red-tailed hawk right there, that red-tailed hawk is just kind of going to jump down and grab you. And so just to kind of also give you an idea of how comfortable red-tailed hawks are in cities and around people, this is a video taken in New York City. And if I had the sound on, you would hear sirens, you would hear all the loud noises of New York City in the background. And this is just a red-tailed hawk enjoying this pigeon on the rooftop of a car. And this is nature happening right here in the city, and these predator-prey interactions. And so that kind of got me thinking about, okay, well, maybe these two animals, the way that they're interacting with their prey is different. And so it might be within the pigeon's best interest to stay close to humans when it's around a peregrine falcon, because the peregrine falcon is not going to come down and come near the humans. And the peregrine falcon is going to catch that pigeon in the sky. And therefore, for the flight initiation distance is a lot closer. But with a red-tailed hawk, if you're a pigeon and you know that there's red-tailed hawks in the area, it might be your best bet to get out of the way, get up in the air, and get away from that red-tailed hawk. So that's kind of what we're thinking is going on here. And of course, we have some ideas about how we could test this in the urban setting. So just kind of some mini takeaways from this. You can get closer to pigeons in areas with higher population density, pedestrian density, and more impervious surface cover and roads and less tree canopy cover. And you can get closer to pigeons in areas with higher peregrine falcon sightings and fewer red-tailed hawk sightings. So what does this tell us about urban evolution? First, urban landscape heterogeneity may contribute to variable genetic and behavioral responses across a single city, and therefore we really must examine patterns at different spatial scales. Second, organisms that depend on humans, such as pigeons, exhibit fine-scale spatial genetic structure that reflect human patterns and distributions, and therefore we really need to sample across broad urban areas. And finally, not all cities are equal, and we really need to understand the historic and ongoing interactions that may be taking place within each city. And so this leads me into what my research is going to be while I'm here at WashU, kind of taking on this environmental justice aspect and environmental racism. So to give you some background on environmental racism, it is a concept in the environmental justice movement, which developed in the United States around the 1970s and 80s. And the term is used to describe environmental injustices that occur within a racialized context, both in practice and policy. For example, in the United States, environmental racism really criticizes the inequities that occur because of white flight that has occurred, in which white people have moved out of urban cores and to the surrounding suburbs. And this term environmental racism was coined by Dr. Benjamin Chavez. Environmental racism is the racial discrimination in environmental policy, making the enforcement of regulations and laws the deliberate targeting of communities of color for toxic waste facilities, the official sanctioning of the life-threatening presence of poisons and pollutants in our communities, and the history of excluding people of color from leadership of the ecology movements. And I think this is really kind of something that has become more prominent and thinking about more, and then kind of thinking about the history of racism and the environment in St. Louis. And I just want to give a huge shout out to a couple of media sources that have really helped shape my thinking about environmental racism in St. Louis. So first, this environmental racism report that was put together by a bunch of different organizations, including people at WashU, that is really informative, and I highly recommend, if you're interested in this topic, going and seeking out. You can Google environmental racism in St. Louis and find the report. And then if you're interested in learning a little bit more about this as well, there's a bunch of documentaries kind of talking about the history of St. Louis and the amount of just kind of environmental injustices that have occurred in this city throughout time. And so for those of us that live in St. Louis, we know that this city is heavily segregated. And to highlight that, I want to show you a racial dot map. So in this map of St. Louis, this is just St. Louis City. What you have is each dot in this map is a single person, and each dot is colored by the major ethnic group that the person identified as in the 2010 census. So we don't have the data out yet for 2020, but so using this 2010 census. And what I personally see is this heavy divide along Delmar. We know this here in St. Louis as the Delmar Divide, that divides the northern portion of the city, which is mostly Black, from the southern portion of the city, which is mostly white. And to kind of bring in some of these socioeconomic variables, here we're seeing again in yellow, this Delmar Divide, that street. And then in kind of showing in dark green versus light green, these different incomes that are higher incomes being darker green. And then here I want to show you lead poisoning levels of infants or of children, and higher lead level poisonings that are really occurring in the northern part of the city, and then a little bit in the southern part of the city in predominantly Black areas. And so what I'm going to be looking at, I want you to hold this in your mind for just a second while I tell you a little bit about squirrels, and then I'm going to tie this all together, hopefully. So y'all have probably seen squirrels. They are native to North America. They come in multiple color morphs. So they come in this grayish red and then also this Black color morph. They nest in trees, but they'll often be found nesting in urban spaces. I had one nesting in my old classroom. I've seen them nesting in cars. You can see them. This nest has been built in the hood of a car. And they're super common in urban parks and suburbs. And so to me, these squirrels seem like this perfect model system to ask questions about how animals are being affected by these environmental injustices. And so what I plan on doing as part of my postdoctoral research is collecting genetic samples, collecting DNA samples, and gathering ecological data about the city, such as canopy cover and water quality, but then also gathering the socioeconomic data. And to kind of give you some clues about what those are, my plan is to collect data such as soil attributes. Where is the lead in the soil? Where's the arsenic? How are lead and arsenic distributed across all of St. Louis? And where are the vacant properties? Where is illegal dumping occurring? We know that illegal dumping is much higher in the northern part of the city. Where has historical racial segregation occurred? Where has redlining occurred? Where have people been denied home loans because of the color of their skin? Where have there been racial covenants where you weren't allowed to sell your home to someone of a different color? And I want to incorporate all of those data into my landscape genetics model. So the goal here is to collect these DNA samples from squirrels, gather some ecological data, and then gather socioeconomic data. And these will be mostly gathered by mining databases, published databases, along with gathering some of my own data based on what's not available. And then using these ecological and socioeconomic data to create a resistance layers. And then all of this gets plugged into Resistance GA, which is an R package that optimizes resistance surfaces using genetic algorithms. And that allows me to then select a model that best determines how my individuals are moving through the landscape and what socioeconomic and ecological barriers are preventing them from moving through the landscape. So this is kind of known as landscape genetics and really helps us determine what's going on and how the landscape might be shaping the movement and distribution of squirrels. So that's kind of one aspect of this project that I have kind of started here at WashU. And then the second aspect that I'm really interested in is how squirrels might be adapting to urban environments. And so I want to show you some pictures here of just squirrels using urban spaces. So here you can see that they've made a little home in between these two buildings where they're using this drainage pipe to get up and down between. I imagine that this is actually a pretty nice little nesting area. It's rather protected and only accessible to these squirrels. And here's a squirrel kind of running along this metal painted fence, which I can imagine is rather difficult to run along when you compare to something like grasping on to a tree branch, which your nails can dig into a little bit, your claws can dig into, which they can't dig in along something like a painted metal fence. And so my thought here is that maybe there's some morphological shifts that are going on. There might also be some physiological shifts. So this is known as Shake Shack squirrel. And this is a squirrel who has learned that it can go into the trash can and take out the discarded Shake Shack milkshake container, pull off the lid, and as you'll see, go and drink up that leftover milkshake. And I can imagine that milkshakes are not what squirrels were originally selected to be eating. And I can imagine that there's a lot more sugar in a milkshake than in an acorn, or kind of their normal food sources. And so one of the things that I want to do is do a genome-wide association study that kind of incorporates things like these morphological variables, taking measurements of morphology to understand limb lengths, and then also taking things like blood glucose levels from the squirrels, taking some feces to understand some microbiome work, and then associating that with their genome in a genome-wide association study to see if we can kind of understand where there might be some selection going on. And my plan is to utilize the St. Louis Wildlife Project transect, which is part of the International Urban Wildlife Network, or UWIN network. And this is a transect that runs from downtown St. Louis all the way out to Eureka, to the Tyson Research Center, and runs basically from this urban to rural gradient that we can then kind of collect all these data on and hopefully associate along this gradient if we're seeing changes in genomes in all these different areas. Well, that's kind of what I have planned on right now. We'll see how much I'm able to get done with this postdoc. I want to encourage you, if you like urban evolution, if you're interested in this stuff, please check out our blog and follow us on Twitter at lifeinthecity_urban evolution, urban evo. We post often. We are trying to keep up on new literature. We post urban observations and kind of give summaries of new research. And if you're interested in contributing, we welcome you to email me, and we can set you up as a contributor if you would like to write blog posts for us. We would be very, very interested. And so with that, I would like to thank a bunch of people that helped me and my funding sources, and thank the Living Earth Collaborative for inviting me to give this talk and to be a part of them, and William and Sasha for hosting me today. I really appreciate you guys. And I will take questions.
Thank you, Dr. Carlen. That was really fantastic talk, really fantastic and interesting research that you were doing. I'm really excited to see your results about the squirrels in St. Louis. That's really intriguing. So we have questions in the chat, and this is from John Birmingham. He asked, were pigeons introduced separately into Boston and New York City?
So we're not sure. We know that there have been multiple introductions of pigeons just in general, because people continue to introduce racing pigeons and fancy pigeons into these cities. And it's possible that they were introduced separately into Boston and New York. But I also, and based on historical literature, we know approximately when they were introduced in the 1700s, but not, it's all kind of, there are some accounts that just say, and there were pigeons, or, and there were doves, and you're kind of trying to guess, okay, well, are they talking, when they say doves, are they talking about pigeons? It can be a little ambiguous. But I would assume that there would be movement going on between Boston and New York, at least back, when it was just kind of the colonizers, the founding colonizers, moving between those cities, since those were two big hubs of colonization on the East Coast. And so it's still kind of surprising to me that those populations are separating out.
Thanks, Liz. Awesome talk as always. We have a question here from Swan Gordon, and first she says, excellent talk and research, by the way, smiley face. And then the question, the question is, it may not have been in St. Louis, but how could disparities in socioeconomic factors possibly inform your old data on pigeon movement and distribution? Do you have plans to consider that as well?
Yeah, so I think pigeons might be harder to look at socioeconomics with, just because their distribution tends to be, because they are capable of moving a lot more. And however, I would love to do this squirrel research in New York. And part of that is because the data sets in New York are so rich, and they really have mapped out where every single tree in the city is. They have I think it's a scaled 1-foot map of all the surface in New York City. And so they have just this very, very detailed map of what the city looks like. And I think that could really start to benefit kind of what's going on, and help us understand what's going on in New York City, at least. In terms of what I'd like to do with pigeons, I would really like to see kind of moving west if the patterns hold true. So is it just that the megacity is so urban that that's what's, they're there, or do we continue to see breaks as we move west? Is there any connection to Chicago or Pittsburgh? I mean, I guess there's some socioeconomic differences between each of those cities, but I think at the fine scale level, even I'm thinking back to my neighborhood in New York City, that the racism in New York City is so block by block almost, that on one side of the street, you'll have incredibly wealthy. On the other side, you'll have much poorer areas. And so that, on the level of a pigeon, I can see a pigeon kind of going back between those two spaces equally. On the level of a squirrel, I didn't see the squirrels crossing those two divided boundaries. And so it might just be the spatial scale, or the scale at which the animal uses the habitat. And so I think there are some really interesting questions there to be answered, and choosing the right study organism, or maybe right isn't the best word, but choosing a study organism that can best help me answer those questions is kind of what I'm targeting now.
Thanks. I think we have time for 2 more quick questions. And this one is from Jacqueline Garnett. She says, hi, Dr. Carlen, sorry, thanks for the talk. She was wondering if you are working with policymakers in urban development or planning, and also in that regard, how urban planning, given your current understanding of pigeons, squirrels, and environmental racism.
Yeah, so my ultimate goal is to inform policymakers. And I personally love cities. I tried living out in the middle of nowhere, and it was not for me. I 100% prefer living in an apartment building in a city. And I think they're wonderful places. And I do want to inform the policy so that we can make cities great places for people and animals. And that doesn't mean just pushing people out of their neighborhoods and completely redoing them, but finding a way to get people to enjoy the nature that we have here. And I think a lot of people forget that something like a squirrel or a pigeon is wildlife. And for those of us that lived in New York City, a pigeon was often the only wildlife that I saw in a given day. It was often the only wild animal that I would ever see. And it connected me back to nature. It reminded me that I'm still in a wild space, that I'm still existing within nature. And I think those moments are really important. And I would happily invite any politician to come out with me. I have, there is an open invitation if anybody is watching this talk and you are a politician or work for a politician and you want to come out with me and do some kind of see what I do. I am happy to talk to you about what I do. I have spoken to our Congress members and our senators on Capitol Hill about the importance of science funding and why this research is so essential to understanding our cities and caring for the people and the wildlife within our cities. And I think we just need this, as I talked about kind of at the beginning of this seminar, we're still in the very beginning stages of studying urban ecology and evolution. And because of that, we're not quite sure what the universal rules are that apply across the board. And so kind of figuring out what those universal rules are necessary before we're, I think, able to make concrete plans that inform policymakers. With that said, I think everybody enjoys nature and wildlife, and that there is a place for nature everywhere within our cities. And the other call that I would make is that we need these data level layers. We need to understand how our water is, what our air pollution levels are, what our soil pollution levels are. And if the city isn't providing that, it's putting a huge onerous on researchers to do that. And there's a lot of benefit that researchers get from this, these data sets that the city curates. I continue to think back to that pedestrian data set where the city was originally curating it for businesses and really trying to encourage businesses to move to certain areas because there's high pedestrian traffic. And here I am as a wildlife researcher coming in and utilizing that data set and leveraging that data set to ask this question. And so I would highly encourage policymakers to use this money to start to figure out things like what is the lead level across the city, and what is air pollution, and where are these
sources of environmental waste. Great. We have one more question. And also I was just, I'm really looking forward to seeing where the Environmental Justice Mapping and Data Collection Act goes that Corey Bush and Ed Markley introduced, because it's basically meeting that exact challenge that you offer. And want to use St. Louis as a test case for that. So it's really exciting thing to look up or to keep watching. Okay, so our last question is from Ansley Petherick, who asks, interesting, back to the pigeon work. Interesting that Providence is a very admixed population between the relatively homogeneous New York City complex and the Boston cluster. Is there something unique about this population, or just IBD gene flow?
Yeah, I don't know what's going on with Providence. So let me see if I can pull up that slide again. It's kind of unclear to me what's going on here. And I think that part of it might just be that my sample size is really low, and only from 2 locations, just because I really struggled to catch pigeons in Providence. And I drove around for days in Providence and just really struggled to find pigeons that were foraging on the ground. It was much less urban than the other cities that I was working in. So kind of unclear what's going on here, and if it just can't separate out because the sample size is so low, or if there's some isolation by distance. I just don't know. That's a mystery. Ansley, if you have any ideas, let me know.
Thanks a lot. Unfortunately, it's time to go. Thank you, Dr. Carlen, for joining us and for sharing your fantastic work. And thanks to the audience for tuning in. We hope you can join us next week in the LC seminar series. Thank you.